the slope.
Yeah. Okay. Actually,
Sorry about that.
We can see it there. Actually.
Really?
Yeah, in bubble on the right.
Is that it?
No,
Or is that
never
the
mind.
base?
That's
That might
the
be the
base.
base.
That's the base. Sorry,
Yeah,
it's
that's
gone.
right here. Yep.
It's floating away.
Well, do you want to slurp the rock pen? Were you interested in that?
Well, there is another one of these corals up and to the right, perched up on a rock. So if Jake wants to try again, there's another one.
Okay.
You'll have to move up.
Yep.
Yeah, it's up and to the right a little bit.
Crispin Little is saying in the chat, this is like picking up a ping pong ball with 2 frying pans. Skills to the pilot. Completely agree.
That's a great analogy.
That is a great analogy. And while everybody's watching you.
Yeah, no pressure or anything.
Yeah.
I think that. Oh, that's great.
Jake, my niece says she's on pins and needles. Will he get it? Won't he get it?
Jake's using the controller for that arm is a miniature version of that arm. So it has the joints and all the...
up here?
Yeah,
Oh,
there it is.
Right next to the Panichia, sea cucumber.
Floating around.
Hopefully it'll get out of the way.
Gonna be
Can I sit down on this rock right here?
It's a fun landing spot.
There's a red jelly off to the right.
Yeah. Cool job.
I know.
If the slope was any indicator before, I'd defer to Bob on that one.
Yeah.
It's hard to tell, but we are on a very steep escarpment right now.
Probably over to the left.
Kim, could you share a little bit about where these samples go? Most Nautilus samples end up at the Museum of Comparative Zoology at Harvard or at the National Rock and Core Lab, the University of Rhode Island, if they're rocks, so they can be checked out by other researchers. But I know these are going to some projects you all are working on. So where will they head?
In this particular cruise, the science party's all from...
For me, ready?
We're all from Scripps Oceanography in San Diego and part of University of California, San Diego. And in this particular instance, the samples will all go for the biology, for the animals, to our benthic invertebrate collection, which is also a museum where the samples are made available for loan, and they'll be catalogued there, and there's an online catalogue where if people are interested they can borrow them. The microbiology samples will end up in Paul's research laboratory freezers. We're starting to think about having a microbiology collection in the long run. We'll see how that turns out. The rocks also will end up at Scripps. We have a rock and core collection as well.
Can you back out a little bit on Argus?
So we really believe in also making our samples freely available for researchers.
I think that's something that's often new for our audiences, this idea that you can check out and borrow samples long after an expedition. So, this is just the very beginning stages of a scientific process by going out and exploring and collecting the samples years afterwards. And you can stay tuned to the Nautilus Live website as discoveries are made and papers are released. Those are updated on the expedition page, so you can see what's come out of expeditions.
Okay. I seem to slide down every time those rocks. Yeah.
Yeah, particularly when exploring the deep sea, we end up collecting a lot of different animals, for instance, and
Okay.
scientists on board might have particular interests, but other animals that are outside that interest get collected. And it's good that those can be available for other researchers to study. And we're really still beginning to understand deep sea biodiversity. There are so many species still to be named. And it takes a long time to name a species. On average, across all of the thousands of species that are named,
All right. Right
those
there.
from the
Ready?
See if I
ocean,
hold there. Yeah.
takes between, on average, 6 to 10 years for each species to actually get named from when it's first collected.
Might have to. Yeah. I'm struggling.
Well, we can move on, Jake. Looks like a very tricky one.
I don't know. What are you thinking? Where would I land right here?
Yeah, switch.
It's not a super important specimen. We have them before, so hopefully we'll see them somewhere easier up the slope.
Okay.
Okay, we'll continue on AJ.
Yeah.
Okay. It's going to be 080 upslope. Bridge nav.
The mushroom coral will live to feed another day.
Step 100 meters, 080.
Maybe it's a good time to just mention the main objective of this project, and this is being funded through the NOAA Oceans and Exploration Research Program. And NOAA is interested in having researchers explore the deep sea, especially in areas like this that are rich in certain types of minerals that have commercial value. So we picked what's called the Southern California borderlands, a wide region off the coast of Southern California that's known to be rich in things called phosphorites and
You can zoom past the arm, Jonathan.
and ferrous manganese crusts.
Who put that on there?
And there are no immediate plans to mine these sites, but there are interests in mining sites around the world that do contain are enriched in these minerals. And so NOAA is funding this project so that we can document what's here to get baseline information about the biodiversity. And so we're doing that. And we're also, NOAA expressed interest in understanding the biopharmaceutical potential of that diversity. And I think their rationale for that is if these habitats were to be disturbed at some point in time, that loss in biodiversity might not only affect the functioning of these ecosystems, but it might also affect some of the value that comes from that biodiversity in the terms of potential new drugs, new pharmaceuticals to treat diseases. So this is really a project of exploration, and we're really just trying to get a sense of what's here and what the biopharmaceutical potential exists for both the larger things, the invertebrates, the smaller things that live in ocean sediments, the so-called micro, macrofauna, and also the bacteria.
Yeah, that's another acester clam there, and we just saw a swiftia. Acester can tolerate pretty low oxygen levels, and we're in a low oxygen area now.
exciting to be out here looking and just doing the basic biodiversity surveys. There's some questions coming in about if we expected to see more life as we went up, but you just touched on it there that we're headed into a lower oxygen area.
Yeah.
Could you talk us through that?
So on the surface levels of the ocean, we have a lot of oxygen production by plankton, phytoplankton, and that provides much of the world's atmospheric oxygen, and a lot of that ends up being dissolved in the ocean water. But when that dies, that plankton and other plankton, it's consumed as it slowly sinks by microbes, and they take oxygen out of the water in that process. So what we see is a decline as we go deeper in the oxygen level. And in many parts of the world, we note here in the Eastern Pacific, this decline can be pretty dramatic in the first few hundred meters, so that around 500, 600 meters we hit fairly low oxygen levels. And so we're now at 730 meters. We'll see, we have been seeing this whole dive as we've come up from a depth of about 1,000 meters. The oxygen has been steadily declining. In deeper water, the oxygen levels do rise back up again. Still not as high as the surface levels, but
More trash.
there is this zone between about 300 and 700 meters where in many parts of the world where oxygen levels hit a minimum and then they are higher both above and below that level. So we're now seeing the oxygen is getting pretty low. There are different animals that are able to tolerate lower oxygen levels, and it's amazing how abundant sometimes they can be. And we may hit a band of brittle stars soon, for instance, where there'll be just tens of thousands of them able to tolerate a pretty low oxygen level, but then we'll go through that to another oxygen level and there'll be other creatures. And then once we hit a certain level, we'll start to see the oxygen levels rise again as we head towards the surface. And so we've been interested in documenting through our transects how the animal communities are changing across depth, across substrates, and in relation to the levels of oxygen.
Do you have a target depth or a target oxygen where you want to set the next colonization experiment?
We do. It's at about 700 meters, so we're nearly there. And that's because we deployed the first experiment this morning at about 1,000 meters. We'll do the second ones at 700 or so meters. Because we did a replicate set of experiments further offshore at another site at those depths as well. So to try and give us a relevant comparison of inshore versus offshore, we are trying to deploy them at similar depths. So in the next half hour or so, you should see us deploying various pieces of wood and rock and even bone. And we'll be leaving those down for about 11 months, and hopefully we'll pick them up this time next year. Oh yeah, move my mask. Yep.
Things are picking up a little here.
For those watching at home, if you do have questions, send them in on the question bar just below the live stream. We'll be answering questions for about 15 more minutes, and then this dive will continue and carry you along through the oxygen minimum zone.
I think I saw a small bubblegum coral back there, little stick.
Looks like we're happening upon some rocky areas, which will be really good for those deployments.
Spectacular glass sponge there with a big king crab sitting inside it.
Wow.
You want to look at it?
Huge, tall.
No, thanks, Jake. We
Okay.
can keep going.
Roger.
Oh, and another one coming up again.
Lots of broken rocks here.
Anyone listening in is a graphic designer. There's a lot of inspiration in the deep sea
Is that another
And-
piece of trash?
and trash.
Trash. Also inspirational to do better.
We've seen quite a lot of trash today relative to the other dives we've done on this cruise, but we're at a relatively near short site. We're not very far offshore from San Diego, and a lot of fishing boats come out here to catch fish, and unfortunately have thrown over the side cans and plastic. And some of the cans we've seen are quite old, more than 30 years, given the designs we can see on the cans, so they persist.
It's a dead sponge.
Yeah.
Right.
Looks like in Argus sonar we can see it starting to flatten out up ahead, and it looks like that matches the bathymetry. It'll be a flatter slope, and hopefully we'll find a good spot for that deployment near AJ.
Yeah, it was a bit of a challenge this morning because it was quite steep for putting out the rocks and wood, and especially the bone cage, so flat will be good. Oh, what do we got here? A piece of...
Whoa.
Quick
It's like
zoom, John.
sonar? What's that?
What does that do?
Come on. Just gonna keep going.
Yes, thanks. That's a piece of Navy equipment that Bob was explaining off mic.
Huh.
To do with sonar.
Huh.
We still have a couple of rocks to collect, but we can't do that until we deploy our experiments. So when we do get to this next site, we'll also be collecting. We need to collect 2 more rocks, 3 more rocks, 6 rocks total. We can put, we could use
2
that for
more.
biology, right?
Yeah, 2 more.
Yep. 2 more essential, but potentially 1 more.
Do you guys want to hear a worm joke?
Of course.
What do you call a worm with no teeth?
Gumworm.
That table
Yes.
looks pretty good.
A
for
gummy
deployment.
worm. Oh, I just heard Kira say that table looks pretty good for
Possible.
deployment.
Yeah, we're just definitely flattening out up here. We
I think I'm
Let's see
getting tugged
here.
a bit.
You can see here we got a lot of bit flatter. The ship's coming to a stop here. I can keep going, or we can kind of start to look around here. We're just around. You see that point, AJ, on your screen? That's.
Yep.
Yeah,
Oh,
we
we're
can
about at it.
keep
right?
going to the mark. It's
Yeah, so you can make your way up there.
Okay.
oh, we're at the mark.
Just about.
Yeah.
We can bring Hercules over to
Yeah,
it.
let's look for a slab of rock, or a cluster of rocks. We'll want to pick up a rock where we put down the deployments.
So how's this look straight ahead?
Yeah, looks good.
Hey, Greg, there's nothing magic about 700 exactly, so if you can find a
Can't quite hear you, Lisa. Can you speak up?
if you can find a rockier area, it's fine to be, little over 700 or whatever.
You don't like the look of this area? It's quite rocky. You'd rather more rocks.
Well, if the sonar suggests there's some more rocks further on, it's okay to go there. But
I think pretty much where we just were coming up, that was the rockiest bit. This flat area we're in now, it's looking like from the sonar, it's going to be patchy rocks like we have in front of us here in Hercules. Probably similar terrain.
Decent boulders right there.
Yeah, like this section here.
okay, yeah, that where you are coming up to,
Yeah.
it's fine.
Okay.
Oh, looks like a good
This is pretty rocky.
table
Yeah.
rock.
Let's use these slabs right where the lasers are. That looks pretty good.
Okay.
Thank you.
The eagle has landed.
So what you're going to see here is the team unpacking a variety of different settlement media, and we can hear a little bit about them as we go. But first off, could you answer, people are interested why you wanted a rocky area to deploy the experiment?
This project is about rocky substrates. And so when Lisa conceived this experiment, the idea was to really get an idea of colonization in relation to rocky substrates rather than sediments. So we have chosen rocks to deploy to see, given there's native rocks all around it, whether various kinds of rocks will be colonized and form similar communities to the background rocks.
Okay.
And if we put them straight onto sediment in a way that would confound the design. And then we're also interested in and added on the idea of, well, what will colonize wood? Wood is a feature of the deep sea that washes out from the land, and there's special organisms that can colonize wood, so we decided to also add on wood. And then bone, when marine creatures with bones like fishes or whales or dolphins die, they fall to the sea floor and they can also be exploited by animals and bacteria. So we're interested in comparing wood, rocks and bone at near to shore and offshore at similar depths. But the major interest really is in what will colonize these rocks and will give us a sense of the time frame, given that they'll be down for 11 months. And can the community that we pick up reach the similar level of the background natural community? And we're collecting also rocks and quantifying the organisms that are living on those. So this gives us a temporal aspect to what can live there. But also
Are
we're
we good
putting
with this
down,
spot?
yeah, this is good. Thanks, Jake.
Okay.
We're also putting down different kinds of rocks to see if the actual nature of the rock will result in different kinds of organisms colonizing them.
Hello.
The float
So
works.
we're going to put out 8 things here. We're going to put out a marker, a float that's got reflective tape on it. Even though we'll take a good GPS reference point, it can be challenging to find these things coming back a year later. So we want to give the pilots as good a chance as possible with their lights to pick up a reflecting target. So we've got reflective tape on these floats that'll be a bit up off the bottom.
Any particular spot for this?
Anywhere there, thanks, Bob. And then if you can just spread around the other things about 1/4 meter apart, each one,
Okay.
not touching each other, but just sort of spread out over those rocks.
It's on the top here.
The rock?
not the top.
I think it's, yeah, that one, yeah.
Or just a grab bag there of treasure.
Yeah, so there's a mesh holding the rock attached to some polypro rope to make it easy to pick up. And
Good.
that's perfect. And in a year or so, when we
Let me sort of
pick
zoom in
this
on
up, the
that.
mesh
See
will,
if I can get the rubber band off.
try and help us
So do we know
keep
which
all
one
of the
that
creatures that
is?
have colonized the rock.
Can't see the
Looks
label.
like one that we collected, so I'm guessing
Can you,
one.
Caitlin or Kyla, make a map of this as once it's deployed?
Yeah, just trying to figure
Thanks.
which one that is.
And I know Kyla's taking lots of screen grabs of what these look like.
I am.
I'll do a
Is that an
rough
SCP?
diagram. Oh, PR.
PR?
Sorry, I'm trying to
PR2. Yeah.
stretch it out a bit.
Yeah, I think it's PR2. We had a quick glance of that. PR... 2. PR2.
PR2.
Yeah, we're going to
Oh,
call it PR2.
it's not moving.
Beautiful. Look at that
That's better.
shot.
Almost.
I like pin it with Mongo.
Science, this is Devin and the lab. It's fine with the rubber bands on it. You don't need to stretch it out.
Okay.
Thank you, though.
You don't want to have any fun.
Now Bob's stuck.
Yeah. Get off.
Snaggletooth.
Almost there.
Yay.
Got it.
And come on, Jonathan.
Coming out.
What's the next one up the pile here?
Yeah, that's the rock. Yep.
SCB. Oh. It's like FM2.
Yeah.
All right, biologist, good question for you coming in. What is the largest animal that could be found here at almost 700 meters deep? What are some of the big ones?
Well, sperm whales can come down this deep if they were hunting squid. There are sharks that live down this deep.
Zoom in on it. Is it okay?
Yes, thanks, Bob. That's good.
Awesome.
In terms of the invertebrates, we're
Come on,
seeing
Jonathan.
very long colonial creatures called siphonophores that are floating by us, and they're tens of meters long. Some of the sponges we've been seeing are up to 1 meter high. Some of the corals we've been seeing have been, not so far today, have also been 0.5 meter or more tall. Some of the king crabs are quite large, a couple of feet across from leg to leg.
W7.
W7.
The hagfish reach a couple of feet long. We've seen sablefish, quite a large fish. We saw one at over 1,000 meters earlier today that was a couple of feet long.
And the deepest
Awesome.
diving marine mammal is the Cuvier's beaked whale. And the deepest recorded dive is 1.9 miles deep.
We actually saw some scour marks from some sort of whale, or possibly beaked whale. That was pretty deep on one of our Davidson dives.
Whoa.
Ooh. How could you tell they
That's
were
awesome.
scour marks, you said?
Yeah, from they looked,
Are you okay with that?
Yes, thanks, Bob. That looks good.
you could say that they were suspected
Yeah,
whale
that's fine.
scour marks, based on the way that they looked and comparing into images of others. What would you like me to label this target?
What was the one this morning? Does it have a label
Yeah, I
from
can
the
find
other
that.
deployment?
Sure, I will find that.
I'm not sure. Does anyone in the lab
It's okay, I have
Could
a...
call up. Devin, if you're in the lab and you're listening, do we have a name for what this deployment should be called?
I have the last one just says NA124.
Is that far enough away?
H1845.
Is that gonna be okay, or do you want it like in the middle there?
Yeah, you can use the same name, but just call it B
B.
or S. Make it S for shallow.
Is that far enough away?
You can use the marker number. This is Devin in the lab. So I believe this is SCB 23, but you can check on the marker.
Okay, zoom out.
Zoom up, Jonathan. Thank you.
Did you record the number, the marker number this morning? Because we could edit that. Well, it doesn't matter. It's okay.
Yeah, you'll definitely have it in several places.
Yeah.
Tied with the observations we're making back there, and also in the...
It should also be written on the basket map.
So here you can see the rocks are different. We deployed the blackish rocks, or encrusted rocks, with a ferromanganese, and then these are carbonates.
You just deployed a marker with the number SCB23.
We did.
Yes.
And you can
Is
name
that
Yeah, I was just wondering what you called the one this
move some of
morning.
these around?
SCB22.
Okay.
Was
Copy
this
that.
one.
Is everything far enough away?
Can we zoom back, please? Just to... Lisa, are you happy with the spacing on this?
They're a little close together.
You'd rather them spread out a bit?
If there's rock there.
Okay. If you,
Well,
Bob, she'd like them a little more spread, please.
Is that still rocky enough?
Yeah, I think
there?
that, yeah, to the right is still rocky.
Or do you want it like way off?
No, no, no. If you can move that one a little bit further over, and then the others can spread out a bit more. And we still got to put out the bone cage
Is that
as
okay?
well. Yes.
Yeah.
You could maybe put the marker off to the left, off the rock. It doesn't have to be on the right. Yeah. Yeah. Thank you. Yeah, I think that slab continues to the right, so we could...
Back.
Like, can we put the cage over?
Yeah, we'll put the
Over
cage
there?
over on the right.
Okay.
Yeah.
Can you describe what's in this cage?
Yeah, so this is a cage, a minnow trap, half a minnow trap with a weight underneath. That's the blue that you can see. And in this we have pieces of pork rib, cow tail, and chicken, various pieces of a chicken carcass. And the idea
How's
is we
that?
want to... That's perfect. Thank you. Maybe the front rock, the carbonate one, could move back and across a little bit.
Okay.
Yeah.
So much for my map.
We don't.
Oh.
Sorry, Paul.
We'll get some screen grabs.
Yeah.
How about over there? Is that
Caitlin?
okay?
And the one that is in the other white rock,
Yeah.
you could now move that kind of into the space where the one was that you just took
Okay.
away. And then we've got them nicely spread out. Yes, that's great. Lisa, how does that look to you?
Bob, I'm not sure if it's possible to get a Argus adjustment so we can get a top-down shot.
that's
of the
fine, thank
experiment.
you.
Okay.
Great.
Looks like it's pretty obscured.
That looks great, team. Nice work.
Can't really
Yeah,
see it
it's kind of obscured.
right there.
Lisa, have you been taking a biology rock from the sites where you're
Yeah,
deploying?
so yeah, we'd like to
Okay,
pick up a
good.
rock here.
Okay, where's that going to go?
Probably in one of the very last starboard bio box, or we also have a forward bio box
Yeah,
face
since
now.
you've got the box open, we can find
Yeah.
a bigger rock to put in the front. How
You
about
want-
that one right to the left of the manipulator? How big is
That's
that?
kind of big.
Big, too big? Yeah.
I think it is. I don't know.
Challenge accepted.
It would be great if somebody could also just draw the orientation of the colonization experiment.
Yeah, there's several maps being made.
Super, thank you.
Hopefully.
With lots of arrows.
Yeah.
Lots.
We've got screen grabs as well.
All right.
Punch in there.
Kyla, can you do one too?
Yes,
You're an
I
artist.
did.
Oh, you did?
I drew one, but I think that
Okay.
we definitely will have to look at all of our
Might
drawings
fit.
and make sure they match.
Whoops.
Oh.
Okay.
Oh, it's a porch rock now.
I think that was too big anyway.
Yeah. What do you think about this one?
Can we zoom in on that?
Coming in. Ooh, scale.
Ooh, that's
Too big.
That's a big one.
yeah.
Yeah.
Cool rock.
Porch rock.
Yeah, the sediment hides what's underneath. Well, we can move. Doesn't have to be right here.
Yeah.
We could find a better candidate. Lisa,
Can you zoom out?
did
Coming out.
you, Lisa, do we have any Niskins yet?
It's pretty disturbed, right?
Yeah,
It's
we'll
really-
do one maybe later when we lift off.
Do you think that before we leave, we could actually quickly zoom into the labels of each just to confirm that we have the labels correct on our drawings?
Let me
If possible,
let-
if it's not
you wanna
oriented
just
towards
kinda-
us.
just kind of rotate so you can get the whole view in sight there.
Yeah, that'd be great.
The vehicle.
Okay.
Thank you.
Yeah.
Or even from here, just maybe zoom into.
I don't think he can't pan over like completely.
Okay.
You just need to rotate just a hair.
Would also be good to get a Niskin as close to around here or where you collect the next rock.
Will do.
Thank you.
Is there too much dirt right now?
Yeah, we're going to have to move to pick up a rock, so we'll.
So for those of you not familiar, the ROVs
Center
are
up the view.
equipped
we
with
can-
6 what we call Niskin bottles, and these are ways to collect seawater. And they're open, and the pilot can trip them, and then it encloses the water. And
You need to go to the right, or we can't pan
Okay.
over there. I know.
so it's best to collect these when you haven't disturbed up the sediment, so you're just getting what's in the seawater.
Looks like a great experiment. We'll be very
Yeah, if
happy
you
for
plant
your-
there.
Thanks, team, for answering all the questions. Gonna sign off here from shore and wish you well on the rest of the dive and a great rest of the expedition. For folks tuning in, tomorrow will be another Q&A session. Check back on the website for when, and we'll look forward to answering your questions and having you along on the dives.
Thanks, Megan.
Thanks, Megan.
Thanks, Megan.
Thank you.
Have
Thanks,
a good one.
Megan. Looking forward to sailing again.
So they want to zoom in on all this stuff.
Rudder.
If you turn too much, you're not going to be able to zoom on that rock.
Well, that's as far as I can go.
Ooh, I think the label's the wrong direction.
That's okay.
I feel pretty comfortable with my
Yeah.
map.
Feel pretty confident. Those are the two
Think you're,
carbonate
driving
rocks,
forward,
CR,
look at your,
SM2. Yeah.
Good.
might stabilize.
The two labels that I didn't get were the wood labels. Did you guys get the
Yeah.
wood labels?
Yep.
Sure.
8 or wood.
8's the upper one and 7's the lower one.
Great,
Yeah.
thank you.
That's it for Zooms, or
Yeah, I think
okay.
this is
So now we want to rock
yes, please. A lock somewhere near here.
and a Niskin.
A rock and a Niskin.
But not in that order.
Could be a Niskin before we settle down again.
Okay. How about those rocks right on the upper
Well,
right?
I mean, the water cleared pretty good if you want to take it now.
Okay. Yeah, if you think it's clear, let's do it.
Yeah, I think the current's blowing pretty good. And
Okay.
it's like we're kind of facing into it. So. What number are we going for?
Number 2.
2.
And this is going to be sample NA124-160.
Copy that.
Watching?
Popped.
Right.
So the deployment's got a number, Caitlin?
The deployment did not get a sample number.
Sorry, what was 159?
159 was the ultrafine sediment slurp.
Oh, I missed it. Sorry. Got it.
So rock.
Was that Niskin 5?
Should have been 2, right?
That was Niskin 2.
Sorry, opposite.
Skin 2.
Second of 6. not the
160.
second to last. Got it. Got it.
I think we want to pick up and look around for a rock, right?
Yes, please.
Definitely lighter.
Like beautiful shot out of Argus right now on sat feed 2. Jake is gently floating something the equivalent size of a Volkswagen Beetle. Or bigger.
See any rocks you're interested in? I don't think we want to go too far away.
Yeah, any, I think we can pick up one that's sort of right underneath you now. One right in front of you now.
Okay.
So I had a question from on my private chat line on my phone, actually from my sister, asking about these red fish because she's tuning in for the first time, and these are called rockfish. There's a number of different species. It's actually a commercial fishery for them, although I don't know if they fish out this deep or not.
Can you zoom in? The rock.
Maybe the more slab-like one, Bob. I'm thinking this one.
Like this one over here? Oh,
A couple rocks?
You think it's 2? Maybe it's...
Yeah. Oh, no.
it's pretty, might go down deep.
Ooh.
Yeah, it's too big.
Slab in the upper left.
Yeah. Yeah, that one upper left is good.
this one up here?
Yes, please.
It's pretty far reach. But yeah.
Stretch.
Well done.
Big stretch. Nice.
There we
Looks
go.
good.
Awesome.
Getting some good screen grabs.
Gently. Whoa.
Where's this one going?
You can go in the forward, either one.
How big is it?
You think it'll go in the starboard?
Yeah, it'll go.
10 by 10?
It would go in a starboard box if you wanted.
Yeah, okay. Let's put it there.
Okay.
Beautiful. The starboard bio
Come all
box
the way
that's
out.
open is the forward outboard box.
Forward, outboard, outboard.
I can just go with the Argus. Don't zoom in too much on the Argus view. I can't see.
Okay.
The forward, outboard, starboard, bio box. Got it. And this is sample ID number NA124-161.
Rock number 5.
Coming in.
And for Paul's sister, those were short spine, thorny head rockfish.
Wow.
So this is 161?
Correct.
Which compartment was that?
Forward outboard. 4 green.
Also known as foregreen.
We have all the different labels. We want to make sure everything's correct.
Is that like a purplish rock or is that a sponge chunk on the bottom there?
Some kind of encrusting sponge or encrusting tunicate, I would suspect.
Cool.
Do we have any other tasks at this site while we're set down here?
What do you make that out to be?
There's a little Swiftia
Swiftia.
you could go and look at.
I wouldn't mind another Swiftia. This one looks a little small maybe, but
Might be 2 there.
yeah.
Is this a slurp sample? Or
Not sure it's going to be slurpable. Get a closer look at it.
Yeah, we're looking at this one and then maybe that one.
Okay. Can zoom there,
Both
Jonathan.
attached to rocks, it might be easier to just grab them.
If they're little rocks, that looks like a
Look
little
like
rock.
little rocks, yeah.
These-
Yeah, let's give it a try.
It's lots of little tiny pink brittle stars.
Yeah, a bunch of them. The sediment on the rocks.
You can see them
They're so
waving their
small.
tiny little arms in the air.
You parked, Jake?
I might be too far away, though. But
yeah, you want to zoom out?
Coming out.
Tilt down some so you can see where your reference.
There you go.
I like that cow pie rock.
It's too big.
For a
Porch rock.
Porch rock. I like the shape of it.
It's an anchor rock is what it is.
Not enough rocks, too many rocks.
dust storm.
Where's this going to go?
Probably in one of the forward bio boxes.
Yes, forward to either side.
There we go.
You should try and keep it down so you're like more balanced. If you're out here, you're too wobbly.
So like this?
Yeah,
Okay.
yeah. I mean, it's kind of tight in there. You sort
Yeah.
of sit a little to the side. I don't know. But if you're out here, it's too,
Okay.
you know, too wiggly, especially when the ship's moving around. So we going for the close in one or the far away one?
I think we'd like them both.
Oh, okay.
Yes, please. They can go in the same box.
You want to be your pan and tilger?
Sure. Oh, there goes my mic.
Oh, you didn't need that anyway.
Can you zoom a little bit there,
Yeah,
Jonathan?
I'm in.
I think your grip force is all the way up.
Oh,
so you might
okay.
want to turn it down some. Try 3.
Pretty small sample. But we'll take it.
Jumping.
See, Bob, this would be a great time for my
You want to zoom in more?
Oh, rock broke.
tough visibility here.
I think, yeah, you might go for the other one and come back to this
Okay.
one.
Come on, Jonathan.
Wide, wide, wide.
Thank you.
The water clarity is pretty remarkable.
What are some of the factors?
Zoom in a bit there. Thanks.
What are some of the factors that impact perceived clarity like this? Because it's a sharp contrast to the water further up north on our earlier expeditions this year, which were very hazy and difficult to see through some spots.
Where was that?
I was thinking up in some of the earlier work up in Canada and, whoops,
Come on, a little bit, Jonathan.
coming out.
Thank you.
Is it
Nice
know it.
grab.
current or depth?
Was that seeps?
It was, yeah.
Yeah, so it can be a lot of flocculent material in the water around seeps because it's a high energy environment.
Yeah, it grabs that.
Nice grab.
Lovely.
Yeah, really nice.
Maybe the other one's too small. Anyway, Paul.
Yeah, we can just go with it.
Yeah, we'll just take this one.
Okay. Tell him to zoom out.
Zoom out.
Put it in the box.
Where do we want this? Front?
The front.
Yeah, either one.
Hey, good.
Thanks, Jake.
Starboard stuff.
Oh yeah, we still got to kick that rock off the porch if you don't need it for ballast. That was not taken as a sample. It just got dropped up there.
Okay, tool tray coming out.
Okay.
If I'm not mistaken. I don't think you guys.
Well,
marked that as
we still need.
Yeah, we still seem a little
weight,
floaty.
yeah,
so.
yeah. So we'll keep it.
Is port side good?
Yeah, that's fine. Thank you.
Perfect. So this is going to be sample NA124-162. And it's being put in the forward bio box port side, which is number 6 white.
Copy.
We're coming in. Did you want the other one, or it's just the one?
No,
No, it's
Okay.
it's tiny.
Say we got about 50 minutes left in the dive, 5-0. And our next target away from us is A, and that is about 340 meters away.
Okay, we're probably not going to get there, but we might get to that next little slope. But
Sure.
before we go, let's do a slope here, just to get another slope done, please.
In a different jar?
Yes, please. In one of the fine jars.
Where are we looking at getting? Just sediment or near
Yeah,
rocks?
yeah, thanks, Jake.
So,
Right there is one of those orange worms, so we could slope
Okay.
all around that. If you could try and get one of those out, would be great. They'll,
sample jar for.
We could
So,
go,
which one's which jar are we going for?
we could go for jar number 2, which is the fine
Number
mesh
2.
jar.
All right.
Great, thank you.
Try not to dig too deep. I think we're just sucking up rocks. And
Yep.
We gotta fish them out of
Roger.
there.
it'll probably disappear on you, but if you can just to the,
Okay.
it'll pull in, but if you dig in, it'll probably get it.
Tight?
Yeah, can you zoom in there? Thanks.
Yeah, it's partly gone in now. It's got this sort of funny mucusy tube. And then otherwise roam around, bit of sediment, bit of rock surface.
Suction?
Ready for juice?
Ready for juice.
Get juice.
Oh yeah, he ran away.
Yeah, they're tricky.
See the end of it right there.
Oh,
I wonder if we got it.
I'd have
I don't
got
know.
it.
I don't think so.
We'll wash that slurp jar.
They're devils, I can tell you.
How deep will they go, Greg?
Could be a good 10, 15 centimeters down.
Oh, wow. Okay.
Yeah, the tube's still there.
Well, I'm not trying to make Jake
Excavating.
This isn't an archaeological expedition.
Take a
yeah.
slurp push core, really.
Yeah, there's already some rocks getting in there.
You can go on the rocks and
Okay.
The bad thing is we suck up rocks and then they get stuck on the hose.
right. Oh no, we don't want the rocks, but the surface of the rock is fine.
Don't reach out too much further. You're gonna get past
Okay.
the max point.
Yeah.
Sample
Danger
number?
zone.
coming out.
Yeah,
This is going
go,
to be
go
sample
back over to the, go the other way.
163 and slurp jar number 2.
Copy.
That rock right in front of you, right there.
Yeah, you could run it over the surface of that rock. Yeah, great.
How have these sediment slurps, have they been yielding kind of successful samples?
Yeah, lots of little interesting diversity of polychaete worms. And some very morphologically different kinds. Quite a diversity, in fact. I'll been taking lots of macro photos of these, so I'll make them available towards the end of the cruise.
Great.